Sure do.
So hi, everyone. My name is Gayatri Iyer. I am a PhD candidate in Dr. Alla Karnofsky's lab at the University of Michigan. And as some of you might know, the primary focus of our lab is developing tools for network-based analysis of metabolomics data. And in that spirit, we recently developed a tool called Filigree, which is a bioinformatics software that constructs partial correlation networks from the data and performs differential analysis. And here I'm showing the workflow of Filigree that we had published a while back. And and in this talk, however, I'm not going to go into too much details of Filigree in itself, primarily because Alla Karnofsky, my advisor, is going to give a talk on Thursday here at 11:45, titled Applications of Data-Driven Network Analysis for Metabolomics and Gene Expression Data. And she'll talk more about Filigree. But kind of taking this theme ahead, the goal of the current study is to integrate metabolomics data with other omics data types in a more data-driven context. And to that end, we sought to investigate sexual dimorphism in aerobic capacity rat models through integrative transcriptome and metabolome analysis. And here we studied F2 generation rats that were generated by crossing high and low aerobic capacity F1 generation rats. And we generated RNA-seq and metabolomics data for these rats. And here we performed both a knowledge-based as well as a data-driven integration of the transcriptome and metabolomics data. So in the knowledge-based approach, we performed gene set and metabolite set enrichment analysis on the transcriptomics and metabolomics data respectively. And then we integrated them in a rank-based approach. So we basically ranked all the enriched pathways and then just integrated the ranks to identify a list of pathways that are differential both in the gene expression module as well as the metabolomics module. And so we identified several pathways that were related to amino acid as well as energy metabolism here that seemed to be important in differentiating males and female rats in both compartments. Next, in the data-driven approach, we looked at the directional relationships between genes and metabolites separately in males and females by performing several regressions. So here, basically, we selected a subset of genes that were both differentially expressed between males and female rats, and that were also correlated with metabolites above a certain Pearson's correlation coefficient threshold. And so we looked at the directional sort of relationship between the two by performing lasso regressions. And so two sets of regression models were constructed, one in which the genes are predictors of the metabolite levels, and another set of regressions where the metabolites are the predictors of gene expression levels. And we visualize these relationships between genes and metabolites in networks. And so here I'm just showing what these networks look like here. These networks are where genes are predictors of metabolites separately for males and females. And as you can see, they look quite different. And then here are the networks where the metabolites act as predictors. And of course, a lot of this is still work in progress. We're still investigating what these networks mean in the context of pathways and biology. And so we're further exploring these gene metabolite pairs that we've obtained from these regression models. But some key sort of takeaway or observations that we've found in this study is that the correlations between genes and metabolites are not very strong, at least in this particular data set. And also these regression networks that I just kind of breezed over, they look very different for males and females. So we're still trying to analyze them a little further and deeper. And in addition, we've also obtained some attack-seq data for the same animals. And our plan is to essentially integrate the attack-seq in these regression networks. And some preliminary analysis, we've identified some transcription factors that seem to regulate the expression of some of these differential genes. And that is all I have. Thank you.